Hi everyone, today we're going to be talking about microbial diversity, and we're going to focus on just a few aspects of microbial diversity, primarily on acellular and prokaryotic microorganisms that will be found in this particular category, because in Chapter 5 is going to be the second half of our microbial diversity unit, so that'll be Part 2. So the breakdown of this particular lecture is going to be talking about some acellular infectious agents, which will include viruses, viroids, and prions. Some infectious agents that we've talked about a little bit already in this course. We'll also follow up with discussing the different characteristics of the domain known as bacteria. Remember I talked about the interrelatedness between organisms in the last lecture that I did, and in this particular lecture we're going to focus on looking at kind of morphological attributes of members that make up this specific domain. And if you think about that classification hierarchy, domain is the top of the hierarchy, so there's domain, kingdom, phylum, class, order, family, genus, species. So we'll look at characteristics, some unique bacteria that are underneath this particular domain, and then also we'll talk about some photosynthetic bacteria, and then we'll also discuss member attributes that belong in the domain Archaea. So remember, bacteria, the domains bacteria and Archaea both make up are comprised of prokaryotic organisms. So there's different categories of microorganisms, as we've talked about before in here. There's the prokaryotic versus the eukaryotic, and in this lecture we're primarily going to focus on prokaryotic cells as well as acellular microbe or infectious agents that fall into this umbrella. And so the types of classifications or groups that we have microorganisms that belong to groups in which there is a cell component or structure that is definitive of the organism. So if something is truly cellular, then it is going to be put in the cellular classification or cellular category. If something does not contain a cell, then it's going to put in the acellular classification. So the members that belong in the cellular unit are going to be bacteria, archaea, algae, protozoan, and fungi. And then those that are acellular are going to be our viruses, viroids, and prions that we've talked about before. Viruses are acellular agents that can infect themselves or inject themselves into the genetic material of a host cell. A viroid is going to be an infectious agent that is part of a virus, so something like a protein that's only common to a specific virus could be considered a viroid. And then a prion, as we've talked about before, is an infectious protein, which when it's inside of a system it causes non-infectious proteins to become infectious or malicious. So cellular microorganisms can be broken down further into the prokaryotic organisms and eukaryotic organisms, and we've talked about that pretty extensively in the last couple of units. And then we also have viruses, viroids, and prions, which are going to be our acellular organisms that fall into that particular category. So when you think about the categories of microorganisms, think that there's those that are comprised of cells and those that are acellular. Those that are cells, we have bacteria, archaea, algae, protozoans, and fungi. And then those that are acellular are going to be our viruses, viroids, and prions. And then remember that the cellular microbes are broken down further between prokaryotic and eukaryotic cells. So let's talk about some acellular microbes first. Now, viruses are the most common acellular microbes that there are. A virus particle is, in its full functional form, is known as a virion. Here's an example of a virion, this entire apparatus here of the virus. Viruses themselves have a pretty specific range, ranging between 10 to 300 nanometers in diameter. Viruses can infect all types of cells. They can infect humans, bacterial cells, plants, fungal cells, protist cells, algae cells, as well as animal cells. So really anything can be infected by a virus. Viruses that are involved in inducing cancer in patients are known to be oncogenic or oncoviruses. I'm sure you've heard of those before, but these viruses cause cancer in host patients. And then one thing to note about a virion is that it's going to consist of a genome that is either going to be made of either DNA or RNA. So all viruses will have genetic material, but they're only going to have one type. That's going to be DNA genetic material, or it's going to be RNA genetic material. And then that material is protected by a surrounding structure known as a capsid, which is a coat of proteins that basically provide protection for the genetic material. And the protein coat on it actually has different packets of proteins or different units of proteins that are referred to as capsosmeres. So anytime you hear the word capsid or capsosmeres, think protein coat, and a protein coat is something that surrounds the genetic material of a virus. So there's a lot of different structures when it comes to viruses. We can have naked viruses, or we can have envelope viruses. So not all viruses will have a membrane surrounding them to protect their genetic material. All viruses will have what's called a capsid, but not all viruses will have an envelope, which is an outer envelope, and that's comprised of lipids and polysaccharides making up the virus. And here's a great example of an enveloped virus here. Let me make this picture bigger so you can see it. So when we talk about the capsid, we talk about these white little protein structures here. And notice how they are basically built to surround the virus's genetic material, which in this instance is RNA. But some viruses have DNA. Virus will either have RNA or DNA, one or the other, nothing else. There's also an enzyme known as RNase, which is enclosed in this capsid that surrounds the genetic material. The capsosmeres that we were talking about earlier are basically the packets of the proteins that make up the capsid. Okay, so this is your typical structure of a virus, and then surrounding the virus you essentially have this layer that's also going to be considered to be an envelope, because the envelope is what's going to surround this particular virus. And again, not all viruses have envelopes, but the envelope again consists of lipids and polysaccharides, and on the outside of the envelope you also have these little receptor regions, which are called receptor binding proteins. And these binding proteins are used as points of attachment or anchors that allow this particular virus to adhere to a host cell, and then once it adheres to the host cell, it can inject this genetic material down into the cell. And it does that by peeling away this outer envelope kind of peeling apart the capsid to release the genetic material. We'll talk more about that in a little bit. But these are the 5 primary properties that a virus has to have in order to distinguish it from a living cell. Again, either they have DNA inside of them or they have RNA, but you can't have both. A living cell has both DNA and RNA. We know that from our studies on prokaryotic and eukaryotic organisms up to this point in our course work. But viruses are non-living, or at least the debate is that the evidence is leaning towards that they are not living, but they will only have either DNA or RNA, but not both. So viruses are parasitic. They cannot replicate on their own, so they have to interject their genetic material into that of a host cell in order to survive and in order to replicate. So kind of another reason why these organisms are not living, considered to be living. So when you have replication, the viruses and envelope viruses that you would think are considered to be a cell, but they're not a cell. It's just an envelope that surrounds the genetic material. In order for viruses to replicate, they will not undergo the process of mitosis or meiosis. So you're not seeing the splitting of a virion into two daughter cells, or you're not seeing the process of meiosis taking place with the virion or the viroid or the virus itself. So instead, that DNA from a virus must be injected into a host cell, and the host cell has to transcribe the DNA into mRNA, and then that mRNA has to be translated into protein, and then that cell will start to demonstrate properties of the virus. So it's very much the cell has to take an instruction from the virus in order to become a virally infected cell. Now, viruses also have an absence of genes and enzymes that are required in order to produce their own energy. So with viruses, you don't see cell respiration and you don't see fermentation. None of those processes that we talked about in chapter 6 and 7 are going to exist in a virus. Viruses will carry enzymes with them, but they're not going to be used for energy production. They're going to be used for DNA replication or messenger RNA construction. And then finally, viruses depend on ribosomes, enzymes, and the metabolites of the host cell in order to survive. So again, the key thing for a virus is to integrate its genetic material into a host cell and have the host cell produce the virus's proteins as well as the virus's nucleic acid or genetic material. And viruses are kind of placed in their own classification. This is dependent on the presence of the genetic material, either the virus having DNA or RNA, but again, it will not have both. This is also dictated by the shape and the size of the capsid, which, remember, the capsid is the protein outer coat. And let me blow this image up so you can see that. So here's an example of the capsid. Remember, those capsosmears are going to be these packets that make up the capsid. So think of the capsid as a jacket itself, but then the capsosmear is one of the fibers of the jacket that makes up the entire cover. And so the shape and the size will also dictate or enable us to classify the virus. The number of capsosmears, so the number of these protein packets that are on here, can also help provide us information with how to classify a particular virus. Obviously, as I said before, the presence of whether or not an envelope surrounds the genetic material of the virus will dictate what kind of virus it is. And then the types of hosts that are infected by the virus will also play a role. Diseases that are produced, cells that are targeted, and then any antigenic properties or immunologic properties, which basically means what properties does the acellular microbe or virus in this case have to allow it to evade the immune system. So all of that is really important information that helps us to classify different viruses. And here's just some pictures of different viruses that exist and are out there. We have the tobacco mosaic virus, and here is a good pictogram of what that would look like underneath the microscope, and here's the drawing of what that would look like. So notice you can see in this image here the capsid that surrounds the genetic material. Can't really stain the genetic material. Remember, these microbes are so incredibly small that we're not going to be able to see them with the naked eye, and we need very high powered microscopes to see them at a very high magnification. And so this mosaic virus is situated here, and again you can see the capsid. Here's the adenovirus. It's comprised of essentially a capsid that's in a helical kind of structure, which differs from the tobacco mosaic virus, which is more rod-shaped or circular in structure. But the adenovirus has all these glycoproteins, which are these spikes that extend off of the capsid of the adenovirus, and those spikes allow the adenovirus to adhere to a host cell in order to penetrate